When you look at an old map from like the 1500s, there is this incredible sense of the unknown.
Oh, absolutely.
You have these highly detailed coastlines where the mapmakers actually traveled, but then you look at the interior of the continents, and it's just, well, it's blank space.
All right, maybe a drawing of a sea monster or the words, here be dragons.
Exactly. And honestly, up until very recently, that is exactly how we have viewed the human body's largest organ. I mean, the skin.
It really has been a massive blind spot in biology.
Right, because we all know it's our armor. It regulates your temperature, keeps the water inside your body, defends against microbes.
Yeah, but for decades, we've treated the skin almost like a uniform sheet of wrapping paper.
But despite a massive global burden of skin diseases, everything from eczema to skin cancer, we haven't actually had a high-resolution map of how the skin's cellular geography actually changes across different parts of your body.
We just haven't had the tools to map that neighborhood-level complexity. I mean, a patch of skin on your eyelid is biologically, structurally, and functionally completely different from a patch on the sole of your foot.
Which brings us to our mission for today. Welcome to today's deep dive. We are unpacking a groundbreaking new scientific paper published in Nature Genetics that has finally mapped the single-cell spatial transcriptomic anatomy of human skin.
It's a phenomenal paper.
Our goal today is to build a high-resolution mental map of this massive organ. We're going to explore how 1.2 million individual cells are organized across 15 different body sites, how they physically change as you age, and this is the wild part, how they completely reorganize themselves during severe diseases.
And to really grasp how big of a deal this is, we have to talk about the technology they use to build this map. It's a method called MERFISH.
Hold on. Let me make sure I'm getting this, because I'm looking at the methodology section of the paper. And it mentions that older studies use something called single-cell RNA sequencing.
Yeah, siRNA-seq.
Right. And the researchers make a point of saying that old method was flawed for this specific task. Why couldn't they just keep using that?
Well, the best way to understand the old method, the RNA sequencing, is to imagine taking a blender to a city to count the residents.
Wait, a blender?
Yeah, literally. To sequence the cells, scientists first had to chemically and mechanically dissolve the skin tissue into a liquid suit.
So you blend the whole city up, and you can say, okay, we have 10,000 teachers and 5,000 plumbers. You know who was there, but you completely lose their addresses.
Exactly. You destroy the spatial context. You have no idea what neighborhood they live in or who their neighbors are.
That makes sense.
But MERFISH, which stands for multiplexed error-robust fluorescence in situ hybridization, it completely changes the game. It acts much more like Google Earth.
Oh, I like that analogy.
Yeah, it leaves the tissue completely intact on a glass slide. The researchers use custom fluorescent probes that they light up specific genes right where the cell sits.
So they aren't blending anything.
Right. So looking at 22 donors, ranging in age from 25 to 83, they didn't just count the cells. They mapped their exact GPS coordinates.
But the paper notes that by avoiding the blender method, they actually discovered a bunch of cells that the old studies literally couldn't find. How do you just lose millions of cells in a sample?
Because of mechanical shear stress. The old blending method physically destroys highly fragile cells.
Like what kind of cells?
Specifically, adipocytes, which are your deep fat cells, and sebocytes, which are the cells that make up your oil glands.
I'm trying to picture why those specifically would break. Is it like trying to sequence a water balloon?
That is a perfect way to look at it. A fat cell is essentially a giant, delicate lipid droplet with a tiny nucleus pushed all the way to the outer edge.
Oh, wow.
So when you apply the chemical and mechanical stress of blending the tissue, that delicate lipid membrane just ruptures.
It just pops.
It pops, and its genetic material degrades instantly. But because MERFISH gently maps the cells inside intact tissue, these fragile lost cells were finally able to be seen and mapped alongside everything else.
That is fascinating. So looking at the supplementary data here, by keeping the tissue intact, the researchers were able to identify 45 unique cell types living in the skin.
Yes, 45 distinct residents.
They found 15 types of stromal cells, so those are your structural architectural cells. They found 13 types of immune cells and a whole variety of surface and lining cells.
And when they mapped where those 45 cell types actually lived across the 15 different body sites, the data revealed some incredible site-specific quirks.
Things that seem totally counterintuitive at first glance.
Absolutely.
Yeah, this was one of the most fascinating parts of the source material for me. There's a specific type of surface cell, a keratinocyte, that the researchers called spinous KC2.
Right, the spinous KC2.
And they found that this specific cell type is mysteriously shared between two totally opposite ends of the human body, the scalp on top of your head and the hairless sole of your foot.
It's so weird, right?
I mean, why on earth would the hairy top of your head share a highly specialized cell with the hairless bottom of your foot?
Well, it forces you to think about the physical demands placed on the tissue rather than just how it looks.
Okay, good. What do you mean?
Both the scalp and the sole of the foot require incredibly tough, specialized epidermal barriers because they are under constant mechanical stress.
Oh, I see. Because your foot bears your entire body weight.
Exactly. And your scalp is stretched tightly over your skull, dealing with friction, UV exposure, and the sheer mechanical tension of housing thousands of thick hair follicles.
So they share a functional need to be tough. The paper mentions they both express a specific gene called SOX9. What is that doing?
Think of the SOX9 gene as a cellular hard hat command.
A hard hat command.
Yeah, when that gene is activated, it instructs the cell to upregulate structural proteins that make the physical scaffolding of the cell much more rigid.
So the body is recognizing the friction in those two specific areas and flipping a genetic switch to toughen up the local residents.
Precisely.
That's wild. And speaking of the sole of the foot, the researchers also found a structural cell, a deep fibroblast, dispersed uniquely throughout the dermal layer of the foot.
Right, the reticular fibroblasts too.
Yeah, and it acts like a specialized architectural support beam. But this raises a bigger question for me.
What's that?
If the foot gets specialized support beams and the scalp gets hard hat cells, does the skin just randomly throw these 45 cells wherever they are needed? Or is there like a master zoning plan?
Oh, there is absolutely a master plan, and it follows what the researchers call a centrifugal pattern.
Centrifugal, meaning moving away from the center of the body.
Right. If you look at the center of your body, your abdomen, your chest, your mid-back, the cellular diversity is remarkably low.
So it's kind of boring in the middle.
It's a relatively simple, quiet cellular landscape. But as you move outward toward the periphery, your face, your scalp, behind your ears, and down your limbs to the soles of your feet, both the sheer density of the cells and the diversity of the cell types absolutely skyrocket.
The paper also points out a really interesting nuance about the limbs. It's not just about getting further away from the chest.
No, it's about the joints, too.
Right, the flexural area. So those are the folding zones, like the pits of your elbows and the backs of your knees. They are significantly more diverse and crowded than the extensor areas, which are the outside points of your elbows and knees.
And if we think about the mechanism there, why would the inside of your elbow need more cellular diversity than the outside?
Well, the outside is just a thick shield, right?
Exactly. But the inside, the flexural zone, is constantly folding. It traps heat, it traps sweat, the skin is thinner, and it experiences constant microabrasions from rubbing against itself.
So that complex microclimate requires a highly diverse, hypervigilant immune and structural presence to manage it.
You nailed it.
So if I'm tracking your logic here, let me just pause and summarize the map we've built so far. We are about 5 minutes in, so let's do a quick recap.
Sounds good.
Okay, bullet 1. We know that by using intact tissue mapping, our Google Earth method, we can finally see fragile fat and oil cells that were previously popping like water balloons.
Yep.
Bullet 2. There are 45 distinct types of cells in the skin. And bullet 3. Your skin is not a uniform wrapper. It gets vastly more complex, diverse, and crowded the further you travel from your torso, especially in areas that fold and trap heat.
Which is a perfect moment to think about your own physical experiences.
Yeah, I have a question for you listening right now. If you're someone who deals with eczema or severe dry skin, have you ever noticed that it almost always flares up in those exact same spots?
Like the inside of your elbows or behind your knees?
Exactly. Why is that?
Because that high cellular diversity we just talked about comes with very specific vulnerabilities.
It's too crowded.
Right. The more immune cells you pack into a tight, sweaty, folding neighborhood, the easier it is for them to overreact and cause inflammation.
Which segues beautifully into how these cells organize themselves. Because even in those crowded flexural areas, the cells aren't just swimming around randomly.
Not at all.
The researchers ran the spatial data through an algorithm to see who lives next to whom, and they found that the skin is strictly zoned into 10 highly stable multicellular neighborhoods.
It's urban planning at a microscopic level.
I love that.
Just like a real city has industrial zones, sprawling residential suburbs, and tightly packed downtown commercial districts, your skin spatially groups cells based on their collective function.
The paper outlines all 10, from the dermal epidermal junction to the sweat glands. But I want to focus on two specific types of neighborhoods that seem to dictate the health of the entire organ.
Okay, which ones?
The first is the deep structural connective tissue, which the paper calls the stroma. The second is the perivascular neighborhoods. Can you break down the difference between the two?
Absolutely. So the stroma is the deep, thick dermal layer. To use our city analogy, this is the quiet, sprawling residential suburb.
Okay, the suburbs. Who lives there?
It is dominated by cells called reticular fibroblasts. These fibroblasts are essentially biological factories.
Factories making what?
Their entire job is to sit quietly in the suburbs and manufacture collagen and elastin, which gives your skin its thickness, bounce, and structural integrity.
Okay, so the stroma is the suburbs where the collagen factories are.
Right. Now, contrast that with the perivascular neighborhoods. Perivascular simply means surrounding the blood vessels.
So these are the downtown districts.
Exactly. They are the bustling downtown districts. They are packed with the endothelial cells that form the blood vessels, parasites that control blood flow, and a massive density of immune cells.
Sounds chaotic.
It is. Everything is moving, nutrients are being delivered, and immune cells are constantly patrolling. It's the logistics, transport, and security hub of the tissue.
And this neighborhood map becomes incredibly important when we look at how the skin changes over time.
Oh, the aging data.
Yes. The researchers decided to use this exact spatial map to study the aging process. But to make sure they weren't getting confused by comparing the diverse skin of an older person's foot to the simple skin of a younger person's back, they controlled for location.
Smart.
Yeah, they looked specifically at the abdomen across 13 different donors of widely varying ages.
It was a brilliant way to isolate the variable of time. And what they observed was a literal demographic shift in the tissue.
What happens?
As we age, the stroma neighborhood, the collagen-producing suburbs, literally shrinks. And simultaneously, the deep perivascular neighborhood, the downtown, expands to take over that physical space.
The actual zoning of your skin city changes.
Exactly.
But what is the actual mechanism there? Do the structural fibroblast cells in the suburbs just get tired and stop making collagen, or something more dramatic happening?
It is much more dramatic. The researchers found that the specific reticular fibroblasts that dominate the stroma numerically disappear. They undergo cellular senescence and apoptosis. In plain English, they die and are cleared away by the body.
So it's not that your collagen factories are just slowing down. The factories are literally being demolished and not rebuilt.
Right. The physical loss of those specific cells explains on a structural level why our skin loses its elasticity, becomes thinner, and produces less collagen as we get older. The residents who do that job have vacated the neighborhood.
That is somewhat terrifying, but also makes complete sense. So, time for our second mini recap. Let's lock this in.
Go for it.
Bullet 1. Our skin is strictly zoned into 10 neighborhoods. Bullet 2. The stroma is the suburb that builds structure, and the perivascular zone is the downtown transport and immune hub.
Yep.
And bullet 3. Aging isn't just cells getting tired, it's a literal population shift, where structural collagen-making fibroblasts in the suburbs die off and the downtown expands.
Which naturally leads to a fascinating therapeutic question for you listening at home.
Yeah, if aging is fundamentally a shift in neighborhood populations, how might anti-aging therapies of the future try to repopulate the stroma rather than just temporarily plumping it up?
Right, because plumping the skin temporarily with a topical cream doesn't fix the microscopic zoning issue.
Which we will touch on at the very end of this deep dive, because there is actually a human trial doing exactly that. But first, if these neighborhoods are so rigidly structured, how do they communicate?
That's the million-dollar question.
How does the downtown talk to the suburbs?
To understand that, we have to zoom in on the most heavily armed downtown district, the superficial perivascular neighborhood.
Okay.
The researchers noted that this area functions as a central military base for the skin, heavily packed with T cells, dendritic cells, and macrophages.
The paper compares it to salt skin-associated lymphoid tissue. But if you pack that many aggressive immune cells into one neighborhood, how do they coordinate without just attacking everything in sight and causing an autoimmune disaster?
They rely on a highly secure chemical telephone network called ligand receptor crosstalk.
Ligand receptor. Can you break that down?
Sure. A ligand is the caller, a specific molecule secreted by one cell. The receptor is the receiver, a protein structure on the surface of a neighboring cell that perfectly fits that ligand.
Like a key in a lock.
Exactly. By looking at the MERFISH genetic data, the researchers could essentially listen in on these chemical phone calls to see who is telling whom to do what.
And they eavesdropped on one very specific, crucial conversation involving something called TNF.
tumor necrosis factor. Now, historically, TNF is famous for being a potent alarm bell. It usually triggers massive inflammation.
Like when you get a cut or an infection.
Right. But the researchers discovered something surprising. In perfectly healthy, normal skin, TNF isn't acting as a fire alarm. It acts as an essential baseline maintenance signal.
Oh, interesting.
The immune cells are constantly secreting a low level of TNF, calling a specific type of structural cell called a CCL19 positive perivascular fibroblast.
Wait, so the immune cells in the downtown area are actively calling the structural cells. What are they telling them to do?
They are telling them to stick around and keep the scaffolding of the military base intact.
No way.
And the researchers didn't just infer this from the computer data, they proved it in the lab.
How did they do that?
They took human skin explants, which is living human skin surgically removed and kept alive in a nutrient dish, and they artificially added lab-made TNF to the dish.
And what happened to the physical tissue?
Within a short period, they saw an actual physical increase in the numbers of those specific structural fibroblasts.
Oh, wow.
It proves that this immune structural crosstalk is active. The immune cells are essentially paying the structural cells to maintain the architecture of their neighborhood. It is a beautifully balanced homeostatic system.
Until, of course, the system breaks. Which brings us to the final major finding of the deep dive: disease remodeling. What happens to this meticulously zoned city when it actually goes to war?
To answer this, the researchers brought in a different type of spatial data. They analyzed skin samples from 5 severe distinct diseases.
Which ones?
They looked at atopic dermatitis, which is severe eczema, psoriasis, hidradenitis suppurativa, or HS, and 2 types of skin cancer, basal cell carcinoma and squamous cell carcinoma.
So we have autoimmune conditions, chronic inflammatory conditions, and localized cancers. I mean, these are radically different diseases with totally different root causes.
Exactly. But when the researchers mapped how the 10 healthy neighborhoods warped under the stress of these diseases, they found shared, predictable architectural changes.
So the city responds to invasions the same way, regardless of who is invading.
Yes, the spatial infrastructure gets hijacked. For instance, across the cancers and the eczema samples, the researchers identified a distinct keratinocyte stress cluster.
What does that look like?
The surface cells of the skin literally panic. They radically upregulate a family of stress genes called the KRT6 family.
What does upregulating KRT6 actually do to the cell itself?
It acts as a physical lockdown protocol. When those genes turn on, the cell rapidly alters its own internal cytoskeleton, making itself thicker, more rigid, and highly inflamed.
It's the cellular equivalent of boarding at the windows and throwing a distress flare into the sky to warn the immune system.
That is a brilliant way to put it. And when the immune system sees that flare, it responds by building something the paper calls a TLS, or tertiary lymphoid structure.
I found this part incredibly cool. Can you explain what a TLS is?
A TLS is essentially a makeshift temporary lymph node that pops up directly inside the skin tissue.
Wait, temporary? Like it wasn't there before?
Exactly. Usually your immune cells have to travel all the way to your actual lymph nodes in your armpits or neck to get instructions.
Right.
But during severe chronic inflammation, like in the cancer or HS samples, the body doesn't have time for that commute. So plasma cells and T cells physically aggregate in the skin to build a forward operating base. They literally construct a temporary immune camp right on the front lines.
That is amazing. And the mapping of hidradenitis suppurativa, or HS, was also a massive breakthrough here.
Oh, the tunneling.
Right. If you know anything about HS, you know it is characterized by these painful, devastating epithelial tunnels that form deep under the skin.
Right. The surface cells basically get confused and burrow downward instead of shedding upward.
And the algorithm perfectly mapped the unique, hyperinflamed cellular neighborhood driving the creation of those physical tunnels.
But perhaps the most profound paradigm shift from the disease mapping is what happens to the superficial perivascular neighborhood, that downtown immune hub we talked about earlier.
Ah, the pan-disease culprit.
Exactly. In almost every single one of these wildly different diseases, this specific perivascular neighborhood violently expands out of its normal boundaries.
It just takes over.
It stops being a quiet security hub and becomes a hyperactive, chaotic domain, pumping out massive amounts of inflammatory signals that flood the surrounding tissue.
Okay, let's do our third and final summary to tie all these complex mechanisms together.
Sounds good.
Bullet 1. We learned that skin cells use chemical phone calls, specifically the TNF pathway, to maintain the structural integrity of their local neighborhoods.
Right.
Bullet 2. When diseases strike, whether it's cancer or psoriasis. The skin mounts a predictable architectural defense. The surface cells lock down, they get rigid, and the immune system builds temporary forward operating bases, or TLS, in the tissue.
Yes.
And bullet 3, across almost all skin diseases, the central downtown immune neighborhood violently expands and drives the inflammation.
Which is a profound realization. It proves that skin diseases are not just random cellular chaos. They are a spatial hijacking of the existing neighborhood infrastructure.
The roads and the buildings are the same. They are just being manipulated by the disease state.
Exactly.
Which makes me wonder, and it's a question for you listening right now, knowing that radically different diseases, from skin cancer to autoimmune eczema, all hijack the exact same perivascular command center, how does this change the way you view the medications or steroid creams you might put on your skin?
That's a great question.
Because we aren't just treating a symptom, we are trying to suppress a neighborhood riot.
And that is the ultimate takeaway from this massive data set. The spatial organization of your skin, who your neighbors are, is just as important as the genetics of the individual cell.
Right.
A cell will behave completely differently depending on what neighborhood it lives in. Therefore, the future of dermatology isn't just about targeting a single rogue gene or suppressing the immune system globally. It is about developing therapies that target the physical neighborhood.
Treating the whole city block, not just the individual house.
Precisely. The paper notes that we might soon be able to develop drugs that specifically calm down the perivascular command center only when it becomes hyperactive, leaving the rest of the skin's immune system intact.
Or, going back to our earlier discussion on aging, repopulating the suburbs.
Exactly. Could we inject healthy structural fibroblasts directly into a depleted stroma neighborhood?
And they actually tried this, right?
They did. The researchers actually point to a recent human trial that successfully increased skin thickness on the soles of patients' feet using exactly that method. Yeah, intradermal injection of specific structural fibroblasts. We are moving beyond just treating symptoms. We are looking at literally rebuilding the skin's microscopic architecture.
It is incredibly hopeful. We finally have the map to the continent. And with that, I want to leave you with a final thought to mull over.
Let's hear it.
The next time you look down at your hand, or you look in the mirror at a freckle, a scar, or a patch of dry skin, remember that you are not just looking at a flat, inert wrapper of tissue.
Not at all.
You are looking down at a sprawling, highly zoned metropolis. You are looking at 1.2 million mapped citizens, constantly picking up chemical telephones to talk to each other, maintaining their rigid neighborhoods, building forward operating bases, and defending your borders. What is your skin trying to tell you today?